Somewhere in the Bay of Bengal, on a small island roughly the size of Manhattan, a group of people are living right now who have had almost zero contact with the outside world for an estimated 30,000 to 60,000 years. The Indian government has drawn a legal exclusion zone around the island. Armed Coast Guard boats patrol the surrounding waters. And still, the people on that island have made their attitude toward visitors absolutely clear, with bows, with arrows, and with a consistency that has not wavered across more than a century of documented contact attempts. In 2018, an American man swam ashore after bribing local fishermen to bring him close. He was killed. Nobody who had studied the history of contact with these people was surprised. The Sentinelese of North Sentinel Island do not want visitors. They have communicated this position with remarkable precision for as long as anyone has been paying attention. But here's the part that nobody talks about enough. Scientists studying the genomes of the world's most isolated human populations, communities like the Sentinelese and their neighbors across the Andaman Islands, the highland communities of Papua New Guinea, the interior peoples of Borneo and the Philippine Highlands, have found something buried in their DNA that was not supposed to be there. A genetic signal that does not match Neanderthal DNA, does not match Denisovan DNA, does not match anything in the reference database. A signal from a hominin that has no confirmed fossil record, no official name, and no recognized place anywhere on the human family tree as scientists have drawn it. And the people carrying the strongest concentrations of it are the ones who have been the most cut off from the rest of humanity for the longest time. That is not a coincidence. That is the most important clue in the entire story. Let us start with something that needs to be corrected before we go any further, because the way most people talk about isolated populations like the Sentinelese is wrong in a way that matters. The phrase that comes up constantly is that these people are frozen in time, Stone Age survivors, living fossils, a window into humanity's ancient past. You will hear this language in documentaries and newspapers and conversations between otherwise intelligent people, and it is incorrect in nearly every possible way. The Sentinelese are not frozen. They are fully contemporary human beings who have been making continuous, active, generational decisions about how to organize their society, how to manage their island resources, what tools serve their needs, and how to respond to outside threats for tens of thousands of years. Their culture has not been standing still. It has been developing in isolation, which is a completely different thing. Isolation and stagnation are not the same. Calling them a window into the past implies they stopped evolving while the rest of humanity moved forward. What actually happened is that they moved in a different direction, a direction that did not involve contact with outside civilizations, partly because those civilizations have historically been catastrophic for every small isolated population that encountered them. The framing error matters for a specific reason: the assumption that isolated populations were primitive leftovers from an earlier era of humanity led scientists for a very long time to treat their genetics as background data rather than the headline, a baseline for studying ancient migration patterns, not a source of active discovery. That assumption has been completely overturned, and the story of how it was overturned begins with a series of genome sequences that produced results nobody was expecting. Getting genetic material from populations that want nothing to do with the outside world is, as you can imagine, not a simple task. The ethical and logistical challenges are enormous. You cannot walk up to a Sentinelese person and ask them to participate in a research study. You cannot explain anything to them at all without risking your life. And even for isolated populations that are not actively hostile but are simply very remote, communities deep in the Amazon interior, highland villages in Papua New Guinea, small island groups in the Andaman chain, obtaining genetic material without causing harm or engaging in exploitation requires extraordinary levels of care. The samples that produced the most significant findings came from 3 different sources. The first was archived samples. In the 1970s and early 1980s, before modern ethical frameworks for research on indigenous populations had been fully developed, anthropologists and medical teams collected blood samples from remote communities during health surveys and contact expeditions. Those samples sat in cold storage for decades, largely untouched, because the sequencing technology to do anything meaningful with them did not yet exist. By the time whole genome sequencing became affordable and powerful enough to reveal what those samples actually contained, the populations that had provided them had in many cases become harder to reach than ever. The archived samples were the only material available. The scientific community spent years debating whether using them was ethically justifiable. Some researchers declined entirely. Others moved forward under careful institutional review, arguing that the information contained in those samples had potential value for the communities themselves, and that leaving them permanently unused meant those communities received nothing in return for having contributed anything at all. The second source was environmental DNA, or eDNA. This technology, which transformed ecological research over the past decade, extracts genetic material not from a person directly, but from the traces they leave behind in their environment. Hair, skin cells, saliva, and other biological materials shed into water, soil, or discarded food remains all carry DNA that can be recovered and sequenced. Researchers working in the vicinity of isolated communities, but not making direct contact, have collected eDNA from water sources, from organic material left at the edges of occupied territory, from beach areas near inhabited islands. The quality of eDNA samples is lower than a direct sample and requires more careful statistical handling, but in sufficient quantities and with sufficiently sophisticated analysis, usable genome sequences can be extracted from them. The third source was voluntary samples from populations that are isolated but not completely uncontacted. Communities in Borneo, in the Philippine Highlands, and remote parts of the Amazon who have had very limited outside contact, but who agreed, through careful community consultation, to participate in genetic research. When results from all three sources began to be compiled and analyzed, a signal appeared in the data that the researchers had not been looking for. A genetic sequence that showed up consistently across multiple isolated populations on different continents, in different geographic environments, with no shared history of recent contact with each other. A signal that, when compared against every sequenced hominin genome in the reference database, matched nothing. What followed was not the dramatic moment of discovery that science is often portrayed as producing. What happened was quieter and in some ways more unsettling. A series of independent findings arriving separately in different research groups working with different data, all pointing at the same anomaly and all arriving at the same reluctant conclusion. There was something in those genomes that did not belong there by any existing model of human history, and it was not noise in the data. To understand how significant this was, you need to understand what scientists already knew going into this, because what they found was not the first unexpected guest in the human genome, it was at least the third. And the two that came before it are essential context. For most of the 20th century, the dominant model of modern human origins held that our species evolved in Africa. Oryx left the continent roughly 60,000 years ago in a major dispersal event and replaced every other hominin species it encountered as it spread across the world. The replacement was assumed to be clean and complete. Neanderthals, who had lived across Europe and Western Asia for hundreds of thousands of years before modern humans arrived, went extinct. Homo erectus, which had spread across Asia even earlier, vanished. The story was a clean line. One population advancing, others disappearing, no mixing, no merging, a complete takeover. That story collapsed in 2010 when the team at the Max Planck Institute led by Svante Pääbo sequenced the Neanderthal genome and compared it directly to modern human DNA. What they found was that every modern human whose ancestry lies outside of Africa carries between 1% and 4% Neanderthal DNA. Not a trace artifact, not a statistical quirk. A real, consistent, substantial signal present in billions of living people right now, indicating that after modern humans left Africa, they met Neanderthals, spent time alongside them, and had children with them. The clean replacement model was wrong. The following year, Pääbo's team sequenced DNA from a finger bone recovered from a cave in Siberia. The bone belonged to a hominin that was genetically distinct from both Neanderthals and modern humans, an entirely new species identified completely from DNA, with no skull, no skeleton, no physical description of what they looked like. The Denisovans. They too had left genetic material in living people, not in Europeans as the Neanderthals had, but in populations across island Southeast Asia and the Pacific. Melanesians, Aboriginal Australians, and certain Philippine highland communities carry up to 5% Denisovan ancestry in their genomes. Two archaic hominin populations, one previously known only from fossils and now confirmed through DNA, one known only from a single finger bone, had both contributed to the modern human genome. The clean replacement story was finished, but it was about to get even more complicated. When researchers with larger data sets and more powerful computational tools began examining isolated population genomes more carefully, they found a third signal. Not Neanderthal, not Denisovan. Something else, something that, when the mathematics was stripped back to its foundations, looked like the genetic trace of a population that had interbred with the ancestors of these isolated communities at some point in the deep past, and then vanished completely. No confirmed fossils, no name, no place in any existing account of human prehistory. Population geneticists began calling it ghost ancestry. And in late July 2026, a research team from the University of California, Berkeley, published findings using a new analytical technique called TRACE, which stands for Tracking Archaic Contributions, via ancestral recombination graph estimation. Instead of relying on comparisons against ancient DNA samples, this method reconstructs the ancestral history of DNA segments within modern genomes directly. What it found confirmed what earlier, more limited studies had suggested. An unidentified ghost hominin population contributed DNA to the ancestors of all modern humans before the major dispersal out of Africa. Berkeley researcher Yulin Zhang stated that the ghost ancestry is present in all modern humans, not only in Africans, and that the unknown lineage diverged from the human family tree approximately 830,000 years ago. Around the same time, Neanderthals and Denisovans diverged from each other. Every person alive on Earth today carries around 0.5 to 1% of genome from this unnamed population. And the same study identified a second, even older signal, a super archaic lineage dating back approximately 1.8 million years, whose traces appear most strongly in populations in Oceania, embedded within regions that carry Denisovan ancestry. That ancient lineage appears to have interbred with Denisovans first, who then passed it indirectly to modern humans. The candidate for that lineage is Homo erectus, among the earliest hominins to leave Africa, one that was living across Asia for nearly 2 million years before disappearing. But here is the additional layer that the Berkeley findings sit alongside. In isolated populations specifically, including Andaman Islanders and communities in island Southeast Asia, earlier research had detected ghost ancestry signals at even higher concentrations than the global average. Genetic study of Jarawa and Onge tribe members from the Andaman Islands found traces of a mysterious hominin whose DNA matched nothing in the available database and coexisted with Neanderthals and Denisovans, but was distinct from both. The populations showing the highest concentrations of these signals are not randomly scattered. They are almost without exception the most geographically isolated human communities on the planet. Andaman Islanders, communities in the interior of Borneo, remote highland groups in Papua New Guinea, indigenous Amazonian populations with minimal outside contact. The pattern holds across populations separated by entire oceans and thousands of miles of continent. These groups did not get these genetic signals from each other. There is no shared recent history between them. They received them from the same archaic sources independently, at different times and in different places, because those archaic populations were themselves spread across a wide geographic range before they disappeared. And then through tens of thousands of years of isolation, these communities preserved what the rest of humanity diluted away. Every other human population on Earth has been mixing, migrating, absorbing other groups, and averaging out earlier signals across dozens of subsequent contact events. In isolated populations, the ancient signals survived precisely because there was nothing new coming in to wash them out. The isolation that scientists once treated as a sign of primitiveness turned out to be the preservation mechanism. The most cut-off people on Earth are carrying the most complete genetic record of encounters that the rest of humanity has almost entirely lost. Now, the obvious question: if there was a widely distributed archaic hominin population present across tropical Asia and possibly beyond during the period when modern humans were expanding and spreading, where are the fossils? It is a completely fair challenge. The answer is more interesting than a simple admission of ignorance. Fossilization is not what normally happens to dead organisms. It is an extreme rarity. The conditions needed to produce a fossil, rapid burial in fine sediment, specific mineral conditions, the absence of scavengers, the right kind of chemical environment to replace organic material with mineral over time, are met only in very particular places: river deltas, lake beds, cave floors in certain geological settings. The overwhelming majority of every organism that has ever lived on Earth left no fossil record at all. The ones we find are the ones that happen to die in exactly the right place under exactly the right conditions. Now consider where the ghost ancestry signal is strongest: tropical forest environments, dense jungle in island Southeast Asia, equatorial rainforest in the Amazon, the Andaman Islands. These are precisely the environments least conducive to bone preservation. Tropical soils are acidic, wet, and biologically active. They consume organic material rapidly and completely. Bones do not last in a jungle floor the way they survive in the dry limestone caves of Europe or the arid sediment layers of East Africa, where the great majority of our hominin fossil record comes from. If an archaic hominin population made its home in tropical forest environments as its primary habitat, its fossil record would be close to non-existent, regardless of how numerous or geographically widespread it was. And there is direct evidence that hominin diversity in exactly these regions was far greater than anyone assumed. Homo floresiensis, the so-called hobbit species discovered on the island of Flores in Indonesia, was a small-bodied, small-brained hominin surviving on that island until as recently as 50,000 years ago. A 2024 study published in Nature Communications confirmed details of its extremely small body size and its deep evolutionary history. Homo luzonensis, announced in 2019 from specimens found in the Philippines now dated to at least 134,000 years old, represents yet another distinct hominin in island Southeast Asia, occupying the region at the same time modern humans were arriving. A 2025 paper in Nature confirmed stone tool evidence of even earlier hominins on the island of Sulawesi. And in late 2024, researchers proposed an entirely new species, Homo giluensis, to account for a group of fossils from Eastern Asia, including specimens previously assigned to Denisovans, further expanding the known diversity of hominins living in the region during this period. There were more of them in more places for longer than anyone had previously understood. The ones we have found through fossils are almost certainly not all there were. The absence of fossils for the ghost population is not evidence that they did not exist. It is evidence that they lived exactly where fossils do not survive, which, given where their DNA keeps appearing in living people today, is precisely what you would predict. The ghost ancestry findings have not gone without challenge from within the scientific community. That is exactly how science should work. The most substantive objection is a statistical phenomenon called incomplete lineage sorting. This refers to the possibility that ancient genetic variants can appear in a population not because of interbreeding with another group, but simply because they were present in the ancestral population before it divided. When researchers see a genetic signal that resembles archaic ancestry, incomplete lineage sorting is always the alternative explanation that must be seriously evaluated. Researchers working on ghost ancestry take this objection seriously. The counterargument is statistical. The two processes, genuine interbreeding and incomplete lineage sorting, leave different signatures in how DNA segments are distributed across the genome, how long the relevant segments are, and how they cluster relative to other markers. The Berkeley trace method was specifically designed to distinguish between these possibilities by reconstructing full ancestral histories rather than making simple comparisons. The ghost ancestry signals identified using this approach are not explained by lineage sorting alone. A second objection concerns the completeness of the reference database. The conclusion that the ghost signal does not match Denisovan DNA is only as strong as how well we know Denisovan DNA. We have sequenced one Denisovan individual from one finger bone in Siberia, plus a small number of additional fragmentary specimens. Denisovan genetic diversity as a population is almost entirely unknown. Some researchers argue the ghost signal could represent a divergent Denisovan group different enough from the Siberian specimen to look like a separate population. This cannot yet be ruled out. But even if that is true, it means the Denisovan category covers a far wider and more diverse range of populations than a single cave in Siberia would ever suggest, which is nearly as significant as a genuinely distinct fourth hominin. What does all of this actually mean if the findings are confirmed? The out-of-Africa model does not collapse. The evidence for modern human origins in Africa is overwhelming, and nothing in the ghost ancestry findings contradicts it. What changes is the picture of what happened after the dispersal. Modern humans leaving Africa did not move through an empty world. They moved through a world already occupied by multiple archaic hominin populations, at least two of which we knew about before and at least one or two more that we are only now beginning to detect. In multiple locations, across multiple encounters, modern humans and these populations met, lived alongside each other, and had children together. The traces of those encounters are sitting in the genomes of living people today. We are not the clean product of a single unbroken lineage. We are the accumulation of multiple ancient mergers, a species that absorbed others partially, incompletely, but measurably as it moved across the planet. Whatever the ghost population was, wherever they lived, however they organized their lives across that wide tropical range, a fraction of them is still here, in all of us, at trace levels, and in higher concentrations in the people who stayed in one place long enough, isolated enough, to preserve the signal that the rest of us diluted and nearly lost entirely. One more thing needs to be said clearly before this ends. The people whose genomes are being analyzed, debated in research papers, and discussed in videos like this one are not data points. They are not representatives of a mystery. They are not a window into any past except their own. They are people, fully contemporary, fully realized human beings with their own social structures, their own ways of understanding the world, their own relationships to their own history. The Sentinelese did not ask to become the world's most famous isolated population. They have asked, repeatedly and with considerable force across more than a century of documented contact, to be left alone. That request has been honored imperfectly and violated fatally more than once. The genetic research conducted through archived samples and environmental DNA operates in an ethical space the scientific community is still working through carefully, with reasonable people holding different views about where the boundaries should sit. All of that is true. And alongside it, something else is also true. The people carrying the highest concentrations of ancient ghost ancestry are the ones whose ancestors stayed in one place in sufficient isolation to preserve a genetic record that the rest of humanity spent 60,000 years diluting away. They are not remnants of our past. They are guardians of a memory so old that it took next-generation sequencing, enormous computational power, and decades of careful population genetics research to even begin to detect it. And what it says, when scientists finally develop the tools to read it clearly enough, is that the human story is longer, stranger, and filled with more forgotten relatives than we have ever been willing to admit. The world's most isolated people turned out to be carrying the world's oldest secret. The question worth sitting with is whether we are wise enough to learn what that secret contains without causing further harm to the people who kept it safe.